So hello there, all the participants. Thank you for attending this session of webinar of RSG-Türkiye. And I would like to remark that we are a group of people who are trying to gather computational biologists through all the Turkey and from the various points of world. And if you would like to join us, be a member of our team, there are several social media accounts of us, and we would like to see you also in our working environment. So today's presenter is Dr. Haris Zafeiropoulos, and the topic of the presentation is Microbial Communities Unpacked. One sample, one network, one association at a time. So the stage is yours, dear doctor.
Okay, perfect. So let me share my screen. Okay, so can you see my screen?
Yes, we can see clearly.
Okay, so hi everyone. Thanks a lot for inviting me to give this talk. So we're gonna talk about microbial communities a little bit. And since like I thought that it's gonna be quite a broad audience in terms of background, it's going to be a little bit introductory. So yeah, if some things you have already heard of them, apologies from the start. And if something else is not very clear, please, it's perfectly fine for me like to just stop me anytime and we can discuss it real time if it is easy for you to keep up. But before we start, I would like to introduce myself a little bit, and like it would be perfectly fine if you would go around as well and meet each other. So my name is Haris. I come from Greece, very close to you, and I'm from a very small village, but then I had to move to big city in Athens to start biology, and then I went to Crete in the south, where I had my master's and my PhD in bioinformatics. And after my PhD, I moved abroad for the first time in Belgium, and right now I'm in between jobs. So hopefully, if bureaucracy allows me, I'm gonna be in UK from next month, trying to jump a little bit in a different field of microbiology, like in the evolutionary perspective. But let's take things from the start. So first things first. The first question, like I was thinking to discuss with you today, is what is a microbiome, right? Like it's quite common. It's quite typical. You may have heard of that term like 1,000 times, or like even in advertisements or well, whatever. So is it like once you like everybody listens to the term, they keep thinking of microorganisms, and this is actually correct, right? Like one of the basic stuff when we discuss microbiomes that pops up in our minds is microorganisms, but these a one very important factor. This is that when we discuss microorganisms, we need to consider that it's not just one thing, right? Like it's actually uh, covers like most of the kingdoms of life. Like you could say, like we have like the prokaryotes, where we have bacteria and archaea. But we also have eukaryotic species like that belong belong uh, to the like the you can have them like as under the umbrella of microorganisms. So the thing with those guys, like even if it is especially for bacteria and archaea, but also small eukaryotes, is that it's almost everywhere. Like in most ecosystems you can think of, you're gonna find some microorganisms. And they are actually can do almost everything. Like most of the most important things to sustain to even sustain life in Earth, we have to thank them for going for that. So when I say they are everywhere, they are literally everywhere. Even in places that most of people would think that life cannot exist, like in super high temperature ecosystems, or in ecosystems with extremely high levels of salinity, and so on and so forth. But when we discuss microbiomes, it's not only about the species themselves. So when we discuss a microbiome, we need to keep in mind that we also have to take into account the environmental condition that those guys live in, the anorganic molecules that they are available, and so on and so forth. And most importantly, that they are not just one species at a time, something that we're going to discuss a lot and then a little bit later. But they assemble as communities. So we don't have to think, we shouldn't think of them like as one species at a time, but as a community at a time. So just to give you like some numbers, because I think like they can be a little bit helpful of how complex the things we're gonna see can be, is some indicative numbers of the biological diversity that we have so far, given the fact that what we have so far is just a very, very small proportion of what is actually out there. So up to now, like Baghdad, one of the most successful, let's say, collections of bacteria and archaea, they have managed to contain in their database, in their collection, 100,000 strains, and this is biological. This is biological information, meaning that they actually have them, like they can, they do have them in their libraries and so on and so forth. From the genetical point of view now, that the difference is that over here we cannot cultivate everything that we have found. The numbers, as you can see, they become even more, even crazier, right? Like you can see that we have, for example, in the genome taxonomy database that we're going to discuss a little bit further later on, we have more than 732,000 genomes that they have been clustered to 143 species clusters. So the important thing to keep in mind here is that in the second case, we cannot, as I already said, we cannot cultivate all those species like in the lab, right? Like you may find something out there with the tools and the technologies that we're going to discuss, but it doesn't mean that you can cultivate it. And it's a very important thing in microbiology is that you can cultivate a species in order to describe it and to have it in a collection and so on and so forth. But this is not the important thing. The important thing over here is that, like, to give you just an indicative number, like how many species like we can have over here. And each of those guys now, they do something different, right? Like in order, like to, if you want to have like, if you think of species like in an ecological perspective, everyone has to be somehow different and do something a little bit different in order to be able to coexist with one another. Otherwise, like species wouldn't differentiate, because like, you need them to differentiate in order to have a new species, right? So most of the bacteria that we're going to discuss a little bit more in this webinar about bacteria and leave archaea and other type of microorganisms a little bit on the side. But what I wanted to mention over here is that every bacteria or even archaea, they come with a set, let's say, of phenotypic traits. That these phenotypic traits can be like they can have a very broad how to say that, how to put it, like they can be they range all the different aspects, let's say, of how a species may live. For example, like what is the pH preference? Like if you like to have a high pH, like in the environment you live, this means that you have certain genes that they can support life in this case. Like if you have respiration, or if you can use sulfur, and so on and so forth. So each species, like each of those guys, of those hundreds of thousands taxa, they have a certain set of such traits. So for example, if we go over here, like sort of giving an example so we can discuss this together. Over here we have we have a stinky friend, meaning that they like sulfur, and wherever you have sulfur in an environment, then you also have something that is quite they have a bad smell, like an egg, like when you leave an egg. So over here, if you see, this is like this is the entry of this guy, of the Sulfobacterium or Erythrotrophum in back type. If you come a little bit over here, then you're gonna see something called metabolic pathways. You see the pathway annotation, and then you can see like how many of the reactions that they are required to do each of those steps. They have been found in the genome of this species. So what you can see over here is like more or less what your species can do. Okay, so if you see over here, they can have like sulfobacterium metabolism, like 100% of it. And this is like an indication of our stinky friend, like this guy that can do this. We know that this is a little bit smelly. So this is why we have our stinky friend over here. So now, as I already mentioned, like those guys, they do not live on their own. You would never find one species or one microbial species or bacterial species like living somewhere on their own. This can happen only in the lab, or in a very extreme case that I would really like to hear. Otherwise, like what these guys tend to do is to assemble communities. So based on what we have seen already, each species has a specific a certain set of phenotypic traits, and this constrains, let's say, the whatever is available on an ecosystem, on a specific ecosystem, shaping a niche for the species. So the niche of a species is like what it needs in order to grow in this specific ecosystem. So based on the niche now of each of the species, interactions among the taxa present in the community starting to form. And these interactions then they shape the community structure, and the community structure leads us to the ecosystem function, meaning like the nutrient recycle that we're gonna discuss a little bit further, maybe, or whatever else this could be. So okay, like I have said already, like quite a lot of terms, like we have microbial species communities and so on and so forth. Like the question is like, okay, why should we care? Like from a more human centric point of view. So like we have microbiomes in as we said, like in almost everywhere. One of these parts is like our bodies. So you have like a role for your digestive system, like in the human gut microbiome, like for your immune system, and so on and so forth. Then you have a soil microbiome, where like it's quite crucial for the nutrient recycling and so forth, and so on and so forth. And of course, like you can have like applications in biotechnology. You can see them like drug drug design, and like you can have like tons of applications like over here. And it's not only like what how it can be like how it can work okay, like because those were cases like where a microbiome is doing what you would like it to do. You can also have a very bad situation out of it. Like if you mess with it, for example, like if you keep increasing the temperature of the planet, then you can have quite a bad how to say that, like counter effect, let's say, because like if you keep increasing, like we have like in the deep sea, like venting microbial communities up to now, like they are very important, like keeping something like 50% of CO2. But if you keep putting pressure on that, then it's going to shift, and then you can have cases where the microbiome actually, like microbes would and microbial communities would lead to more and more desert-like cases, let's say. So last thing, let's say, just in order of context, it's like we have microbial species, meaning they are super small. You cannot. It's not an easy thing to even measure them, and this is why, like, we have many different ways in order to try to assess the present, how much of a species you've got somewhere, like and even if you have a single species and you know that you know what species that is, even then it can be quite a hard task to measure the density of that species on your cultivation. Even harder to distinguish if the cells that you've got, they are live or dead. And the more species you have, the harder it gets to get this to get such numbers. And this is why, like, one of the things, like in terms, like jumping a little bit in the bioinformatics world, let's say, one of the things that I wanted to show you today is that over the last 3.5 years now, we have been developing a repository, you may say, called the mGrowthDB, where you can store, but also query and search and compare growth data from microbial community. So what you can see over here is like experiments from an already published study, where you can see all of those different species over here, right? And what was like their growth under the environment and under the medium that it was set in this specific study. If you come over here, then you can link it to the publication and get the medium from there. And you can have that also in the case where you have more than one species, like in a mixed culture. So if we would come over here, let's say, then you would see like what is the what is each species density when you cultivate them all together. Okay. So, however, as I just said, this growth depends on the medium, on where your species live. Some of the factors that affect your growth can be like, of course, the nutrients that are available, the pH, the temperature, and this is just a very small list out of them, right? And here we have like like more on the on growth DB if you would like to give it a shot. And now, like, given that we can measure their growth and so on, we can start, let's say, having a look at what we discussed earlier and we barely introduced as interactions. So we have a lot of different kind of microbial interactions that we can categorize them like somehow, let's say, in positive and negative, meaning like if one of the two taxa, if we think of pairwise interactions, benefits from the other, or the other way around, or if one guy actually harms the other. And the ways to do that, they can be quite a lot. So you have like one of the most typical things that you would see in microbial communities is what you have over here as horizontal gene transfer. So this is very common in bacteria and archaea. So you have like parts of genes, or maybe genetic regions larger than just a gene, that they would be transferred from one species to the other, giving to the second the ability to do something that previously it was not capable of doing, right? Then you have what we're going to discuss a lot about it later on with metabolic interactions, where one species produces a metabolite, they secrete it on the environment, and the other guy, another species, can get it and do something with it. But you also have like many different other ways to interact, like worm sensing is a very nice, like very important way of type of interaction, where you have like actually chemical signals working as a language between among cells of species and so on and so forth. So now, like, since we have discussed all these introductory terms, let's say already, the idea is this, right? Like the idea is like we are bioinformaticians, we are computational biologists. So the question comes like this: like, how could we study the ecology of a species, or even of communities, of whole communities, starting not from the phenotype itself, let's say, but from the genomic information that we can get in a quite easier way. Okay, because like with modern technologies, especially when it comes in sequencing, like we have access to massive data sets that we can explore. So the questions that we are interested in studying is like who lives in an ecosystem, right? Like what taxa we have in a specific ecosystem in a community. Where do they live? Like what are the specific conditions of this community? They meet the environment, the medium, the nutrients available, the pH, and so on and so forth. What they can do, meaning what are the phenotypic traits that we discussed earlier, like of each of the species that they live over there, and how they interact. Like, okay, since we know who lives there, we know what each of these guys can do. How do they interact? Like, do they have positive? Do they have negative interaction, and so on and so forth. So this is like in this slide, like it is like the most of the things that we're gonna discuss from now on. Like we're gonna see like a little bit of the technologies that you can use to get an idea of who lives over there. Like we will see how can we figure out like what each of those guys can do. And then we're gonna try to discuss about their interactions. But before we continue, I would like to ask you if there's any questions so far before we jump into the next. Anyone?
If you would like to ask a question, you can unmute yourself or write in the chat box.
Yeah. Okay. So in the first question is like the who, right? Like who lives over there. So one of the most typical things someone can do is like to just get some DNA, right? So like there's especially if you like natural ecosystems, you know, like marine sample or like sediment. We have a question. Okay, so the question from Gok, if I pronounce it right, sorry if not. We're gonna discuss it later on. It's about like how like how confident can we be in the context of reverse ecology. We're gonna discuss it like later on and how to discuss about if you have a strong or a weak interaction. Yeah, because we need some more concepts to discuss so everybody can get it. So from an environmental sample now, you can have like some DNA-based profiling methods that the most the most well-known, let's say, is like you can distinguish it as in amplicon sequencing and metagenomic sequencing. The basic difference in that is just that you have one target gene in the first case, while in the second case, you just go for everything that is out there. So in the first case, you're going to have the one target gene is not one gene that can be whatever. It needs to be a gene that is quite diverse among the different species, but it is quite robust and quite the same within a species. So in bacteria, this is the 16S rRNA that you may have heard of. That works like as a very good proxy of this diversity among the species, but it stays similar within one. So using one of those technology methodologies, and taking quite a lot of time to do the experiments and so on. What you end up with, let's say, is something like this corona plot over here. So what you want to figure out, what this technology will allow us to get a glimpse of is like the species that they live in a community, and the number of them it can be quite big. This is why I wanted to show you this, because like you may end up with hundreds of species like living at the same time in a community. In some cases, even thousands. It's you can see cases where you have more than 1,000 species of taxa that they live in the same sample, forming communities. So when you have this information, like in order to get this information, these different techniques they bring quite a lot of limitations, but we're not going to go that way today, right? So the important thing is that using those, you can get very important very important characteristics of your community. So we have some indices and some metrics in order to quantify how diverse a community is, or how the different species within a community distribute, like the abundance of each of the species you've got over there, because it's not only what species you have, but also how much of the species you have. You find it in your sample, and then you can have either, like, as you see in the right plot over here, like an actual, let's say, a more concrete number, or you can have it as a proportion. And when you have sequencing, then it's always like this, right? Because your sequencer, your machine can only measure a specific number of reads. So no matter how many cells you have in a species, your sequencer is going to read the same number of reads. So because of that, we have this leads us to one of the most basic problems we have in microbiome data analysis, meaning the compositionality of our data. So you should consider that this chronoplot over here is actually only one of those bars in these plots over here. So if, let's say, this is one sample, it would be SC13 over here, let's say, right? And then you have alpha diversity discussing what is the diversity within the sample, within the community of specific sample. And then you have beta diversity, trying to discuss how diverse your communities are between your different samples. So if you want to discuss how different SC13 is from SC25, you would go with beta diversity and indices discussing that. So since we now know, let's say, we have a brief idea of the species of the communities, of the members of the communities, then we would like to see what they can do, right? So in order to see what they can do, based on DNA, what we want to do actually is to annotate the genes that we found there. So that means that this kind of information, we can only know of that if we have shotgun metagenomes. Because if we would have just the amplicon, then we would only know who is there, and we wouldn't know anything about what they can do. So now, if we go back, sorry for the back and front, just to make it a little bit easier. So in the amplicon case, we would only have this part over here of the whole genome of the species present. But with the metagenome, we can have the whole thing. right? So now we can get those genes and annotate them. And we can do that in two ways. So we can do that in a way where we consider our sample as a superorganism. So all genes come from the same guy, let's say, and then we measure the genes, we annotate the genes and we quantify their abundance as it was one species, and we check what they can do, as you can see over here. Or we can first do something else that I'm gonna show you just next, and see and get the same information, but for each species that is out there. So the amplicon analysis is like you can find just by metabolic coding as well in the literature, and it is as I said, like one gene that if you first amplify it with PCR or whatever, and then you sequencing this gene and you get your community, or you can have the short-term metagenomes, and as you can see in this very nice visualization, you can build contigs that these are like very long parts of DNA parts, and then being them. So you reconstruct the genome, the whole genome of a species present. So the idea here is like, and the quite interesting thing is that you have this thing over here that is so rich over here that you don't know what color they have in the beginning. You don't know anything. You just have parts of sequences, and then you start building longer and longer parts, building your contigs, which you can then combine and build the whole thing you call a genome. You call a MAG in this case. Okay. So once you have that, once you have reconstruct the MAG, then you can annotate the MAG instead of doing the superorganism thing that I told you earlier. So for example, in this case, we had our initial sample was a microbial mat from a hypersaline swamp. And what you can see over here is like we had just a very short number of samples. We had just something like 6 samples, if I remember correct. It's like every sample is one of the circles around, like the purple one, let's say. So you have the orange layer, the combined 3, 4, 5. Yeah, you have 6 samples over here. Okay, and those guys that they built the phylogenetic tree you see in the center. They were the mags that we were able to reconstruct from those 6 samples. So if I'm not mistaken, the number is something about like 180 something. The craziest thing of all with these mags over here is that when we try to find them in GTDB, in this database that I showed you in the beginning, when we were discussing the numbers of the tags that we have available, only 4 or 5 out of them, they were actually mapped in a species in GTDB, meaning that everyone else, it is a potential new species. Okay. At least from the genetic point of view, right? From a genetic point of view, and we can be quite confident that we were right about that, because you can see, like with this green star over here, that almost all of our mags, they had a very high completeness score, right? Like they were quite. It was not like just a part of a genome or something. And now, like, the important thing that I would like you to keep from this is that now that we have a MAG, meaning that it's like we have a genome of a species that we don't have in a collection, but we have managed to find it out there in an actual environment, we can annotate them directly. So we can now have an in silico discovered species, let's say, and annotate this one and then see what this guy can do. Sorry, we have some questions that I just saw. And they are quite a lot. So give me one moment. Gigit says, how difficult it is to understand molecular interaction from shotgun metagenomic data. Gigit, may I ask you to specify what you mean with molecular interaction? If you want, like, just unmute yourself so we can.
Yeah, sure. I'm new at this, and my question might be silly. So my thing I'm trying to say is the functional profiling, actually. How can we deduce that data from shotgun metagenomic data? Is it possible? How difficult is it?
To get what? Sorry, to the functional?
The functional profiling.
Okay. Okay.
Uh-huh.
This is like now it is quite straightforward to have a functional notation of a metagenome. It's not that hard, but you need to always keep in mind, and this is something that I tend to discuss a lot later on, that you can only annotate, like when you annotate something, it can only bring you things that they have already been recorded as such. So like if you have like a database with 100 genes that work as your reference database, and you bring a new genome to check, it's going to scan, your algorithm is going to scan over those 100 genes. But if your genome has a new gene that is not in your reference database, then you're going to miss it. Right. So it's quite straightforward to do the first part, but we have to keep doing it again and again and again, let's say, in order to update our annotations based on the new findings that the molecular guys find in the lab. Because like we still try to annotate genes that we don't know what they are doing. So until now. So for Meltem says,
thank you so much. It's clear for me now. Yeah.
Of course. So Meltem says, from the microbial database, how is it possible to assess individual metabolic activities of each strain in a microbial community? Others based. Okay, that's a really nice question. So it's not just from the genomic data. No, of course not. You can if you have one species in your community, it's quite easy. Like you go with something like called HPLC. And then you can measure, like the acetate, the butyrate that you have on your medium, and so on and so forth. If you have a community, this is when things become much, much harder, because you don't know who produced what, right? So then in the database, you would have, for example, how much acetate you have in your community, but you wouldn't know who produced that. But what you can do, at least, let's say, is that if you have a time series, if you have an experiment that it had, like 5, 10 time points or whatever, you can somehow at least check the growth of each of your species with respect to the concentration of the different metabolites you have over there. What I'm trying to say is that, for example, if you don't have any a specific metabolite at all, and at some point you have a species that starts growing, and at that time point this metabolite starts to increase its concentration as well, then it is quite fair to think that maybe this species actually secretes this guy. But this is quite hard, and you have to do other kind of techniques if you want to find out who is producing what. And then you say, is it possible to detect horizontal gene transfer across strains with marks? Okay. I think it is. Actually, to be honest, this is one of the stuff I'm supposed to check in the future months. But I think that you can do. I think that you can do. I don't know if you can. I don't know if you can do it in all cases, but I think that you can do. But this is also a very good question. We can check it together later on. And then some genomes we find in silico are from microbes no one has grown in the lab yet. Yes, exactly. How confident can we be about the functions or roles, and what could go wrong if we assume too much from just the sequences? So, decent again. So, as I said, we can be sure in terms of the things that we already know that they work in a certain way. So, like if for example we know that a gene is doing a specific thing, then it is quite okay to say that okay, the mark that we reconstructed, it can do this. The most important thing that we do not know with these techniques is what is not already in our reference databases. I think, at least in my opinion, that I think that the most hard thing is that if you don't know, if your species, if your mark has a gene that has not been annotated before and it is not in our reference databases, then when you're gonna try to do what we're gonna do next, then you will not be able to have that, because you need for people to still go to the lab, and take one gene at a time and figure out what it does, and add it in the reference database so we can then exploit their work, let's say. Okay, so this was a good time for the questions, actually. So over here, I have some links to some other presentations if you are interested in this kind of analysis. So if you work with metabolic coding and amplicon stuff, you can have I think it was a summer school over here. If you want to do some shotgun, I think that your guy, Dr. Eren, he has done a super important work on the field. So, by the way, let me share with you the link over here. I'm just putting the link of the presentation in the chat. So, like, if anybody wants to jump in, and you can click on the links and have that over here. And besides that, I also saw that Professor Nagarajan had a talk on modeling complex microbial communities from metagenomic data in another webinar of your community, like 3 years ago. So you should also, like, if you're interested in the field, you should also have a look in this talk over here. It covers quite nice the generalized local Volterra modeling, that it's something very trivial in the field, that people will keep doing it for many years now. And I think it's a very good stuff. place to start. So before we go to the next, let's say, topic, based on the marks that we just discussed, I wanted to have another short intro in the genome scale metabolic models, because we're gonna
Professor,
see it
sorry to
yes.
interrupt your presentation, but we arranged the time schedule like around, is it 15 minutes more is okay for you? Because like the schedule is arranged as such.
Okay.
Is it feasible?
Yeah, for me it's perfectly fine. Like because like we're doing the questions at the same time, so I missed the time. Yes.
No problem.
Okay. I will try to move on a little bit faster, so I don't keep you guys like more than I was expected. I was supposed to. So like given you have a Mac, okay, like this is the what we're discussing. Given you have a Mac, then you can have a genome scale metabolic model. This is not something more than a mathematical representation of your genome. So you annotate your genome. You have a list of the enzymes that you have on your genome, and based on that genome, you can get the stoichiometry of the reactions that they can go for. And since you have the stoichiometry of that, then you can start having mathematical models of what is needed in order to do something, and so on and so forth. And again, like it's not the main thing of that webinar, but if you are interested in that, here is a link that you could go for it. So given now that you have the community profiles that we were looking at earlier on, the question is like, okay, how can we start figuring out the interactions between the taxa that I have in my sample, right? This is the question. So concurrence analysis is like a thing that it has been widely known like for more than 50 years now. In whole, not only in microbiology, of course, but in all fields of ecology. The idea is like, okay, that if someone cannot stand the presence of someone else, then these guys, they avoid each other, they have a negative association, let's say, and the vice versa. If two guys like you find them like together, they can coexist. So maybe this means that they benefit one another, and so on and so forth. So here, like over here, is like some basic history of the idea. For us, what we have now is that we have the shotgun and the amplicon data. So we can use them, to figure out such type of associations and come up with networks like the one you see over here, where for nodes you have some taxa and your edges are like associations between these taxa, right? So there are several software tools in order to come up with networks like that. The one that I have been using in terms of the software I was supposed to discuss about today, but I don't know if we're going to make it on time. It's called FlashWave, and it's quite nice because it uses a conditional independence concept. I'm skipping out the details of that just to have a glimpse, to give you a sort of interpretation, let's say, of how someone should understand such a network. So like, in this case over here, we have three nodes, like a network of three nodes and three edges, like very, very simple case. And in this case, you would have like a positive, let's say, association between ASV20 and ASV29, and ASV20 and ASV26, and then you would have a negative one between 29 and 26. So what does that mean actually? Okay, so you have just a number, but and this number, would range from -1 to 1. Okay. So this doesn't mean anything further that your guys, they were found together or they were not found together. Like when you find someone, you don't find the other. So they don't mean anything biologically. It's just a statistical score. It's a statistical weight. This is the important thing to remember. They can be just like this association can be even 1 or -1, like like as much as possible, and it could be the result of something else that happens at the same time, and it just have the same outcome. For example, like let's say you have two guys that they only live with a pH above 8 or whatever. So you always find them together, or you don't find them at all in your samples, because like you have just a bunch of samples with pH over 8, and all the other samples they have pH of 6 or whatever. So you have a very strong positive association, but they don't actually interact. It's not an interaction. You get my point. It's just an association, because it just happens that they like high pH. Okay. But what, like as we said earlier about the type of interactions that we can have, one of those are the metabolic interactions. So you can have a very nice introduction on those, like in this paper over here. But what I wanted to discuss with you today is like in the concept of the metabolic interactions, you may have complementarities. So what I mean with the term complementarity is that you could have two species that one guy can do something metabolically that the other guy cannot. So the first guy shares something with the second in order for them to actually go for the process, go for what they cannot do, right? So one type, let's say, of complementarity is what we call pathway complementarity, and you could actually think of that somehow like in terms of the horizontal gene transfer we discussed earlier, because what you have over here is that like the potential beneficiary gets a genetic loci or like an enzyme from the other in order to actually go for a metabolic process that on its own the potential beneficiary cannot go for. And then we have the seed complementarity, and this is why I discussed the metabolic models earlier on. So in the seed complementarity, you don't just keep the set of your genes, let's say, but you check the whole metabolism of your species, of your mag or of your genome or whatever. So after you see the whole topology of your species, then you can figure out the metabolites that your species can actually use to do something, but it cannot produce on its own. So they would need to get it from their environment. So for example, if A over here, the box A is one species, then you can see that the metabolite A can be used in order to become metabolite B, and this would be metabolite C, blah, blah, blah. But there is no enzyme, there is no reaction of your species that can actually build metabolite A. So the species needs to get metabolite A from their environment in order to do and all the other processes. So in the seed complementarity approach, metabolite A would be a seed for species, for the potential beneficiary species, and a potential donor may provide metabolite A, species A, in terms of a metabolic interaction. And then if you have that, for example, let's say you could have something as you see over here. So your species, your potential beneficiary, was missing those two guys, the red dots, metabolites. And if you have that, then if they would get it from potentially donors, then they could have like the B6 metabolism like complete. Let's say. Okay. So the idea now of the software that I was like to discuss as a last thing called Microbial was like that once you have a coherence network, you end up with something we call hairball over here. So you have all those associations that they are just statistical, some statistics that you cannot argue about those like a lot. And the question was like, okay, can we do something in terms of having a better idea of those associations, like if something is an actual is more actually and is more possible to be an interaction or over some others. So the idea was to build a software that combining like the phenotypic traits that we can annotate to our species and some network clustering and metabolic complementarities, they could give us a better idea of those associations they could help us understand a little bit further the associations predicted from the coherence network. So for example, what we do in Microbial is that we have a set of traits like the ones you can see over here. We have like something like about 40 traits that we annotate the nodes of network over there, and then you can have we do network clustering on those, trying to figure out if some phenotypic traits are more enriched or depleted in one cluster over the other. And we also have the metabolic complementarities that I just introduced to you. And we have done that in all the pairwise taxa of the genome taxonomy database, not in this version, but in a previous one. So we had over 34,000 representative genomes. The genomes you see over here is like the whole number, but it's not the representative genomes. The representative genomes are now something about I think like double of this number, but doesn't matter. And we have more than 1 billion of complementarities in disclose and so on. And for each genome, of course, we have a metabolic network. So the workflow, like of our software is like this. You have an abundance table that comes from your amplicon data or from your shotgun data. You build coherence network with FlashWift if you don't already have one, and you're trying to map your species to the GTDP species. And once you do that, then you get the annotations or the pre-calculated annotations from Microbial database in your network, and then you have like a Microbial annotated coherence network. And in order to do all those things, we have developed developed Cytoscape add-on. Cytoscape is a very well-known software in order to work with networks. So we have built an app for that. And like once you have that, like you can have like your Microbial annotated networks in Cytoscape and have like visualizations like you see over here. Like so, for example, when you click a node, an ASV or a MAG or whatever that is, then you can see like the phenotypic traits that were assigned to that. and you can also see like the complementarities that were predicted. And what is very nice over here, like for you to think, is that if it was not for microbtag, this pair of AHVs would only have the blue edge, right? Meaning that they would have a positive statistical association without any direction. This is what we call an undirected network. But now you see those extra edges that they have an arrow, meaning they are directed, and this is what you should think like, okay, in one case we have species 215 being a potential beneficiary species, and the other guy is a potential donor, and in the other cases it is the other way around. So using this approach, you can come up and see your networks over like from a different perspective, and you can come up with what we call hypothesis generation. So all of this approach ends up to a new hypothesis of what may happen in your community and does not prove anything. Okay, so this brings us to a question that someone made earlier, that how positive we can be. We cannot be positive at all. You should get this new hypothesis, go back to the lab and check if what was predicted makes any sense or whatever. And you can, last thing is that you can do that not only with our genomes from GTTP, but if you have shotgun genome and you build your own mags, then you can run the whole thing using your own genomes. And we have a demo if you would like to give it a shot. And over here I have some nice papers of that they are related with what we discussed today. And just like a small add, if you are interested in things like that and you work and things like that, or you know anyone that works with metabolic interactions and so on, we have a collection in the Java journal that it would be nice, like if someone would like to submit their work over there. So like these are the people that they had a quite important role in the microtag development, and thank you very much for your time. Sorry for taking a little bit longer.
Thank you for the precious talk, and also thank you for reminding us another seminar of ours from RSG-Türkiye. We can continue with question and answer part, and I can take 2 to 3 questions because of time limits. I'm sorry. So if you would like to ask a question, you can unmute yourself or you can ask through the chat box. Okay, I might ask, is there any kind of recommendation of yours for the new beginners of this field, or for the people who are going to enter this field of research?
Meaning like
Yeah, like
what
yeah, you mentioned cytoscape and other methods, but is there a certain type of knowledge, or I do not know, is there any kind of topic they will be specialized on or take importance?
that you should be, you mean like something that would be to worry about, or something that it would be nice to go for, like as a
Yeah,
new,
the second one.
the second one. So yeah, of course, like for the second one, like we have like a lot of stuff that we like, that they are really nice to consider. If you think of metabolic complementarities, then one of the most interesting thing is like the evolutionary perspective. them, like how two guys they decided, like, no, I give you this, you give me that, and you could have a lot of experiments to check like how this arises, let's say. And then in terms of like some extra features that you could have like in approaches like Microbial and so on, it's like if you think of the slide with the different interaction types, you could start getting one type at a time, adding it in your in your software, right? So, for example, what we are doing now is we are trying to add a channel in our annotation about the quorum sensing. So, if you know that species has some chemical signals that it can send and some others that it can get. Then if you find those two guys associated and you see that they have they have the chemical signals for quorum sensing to work, then maybe the actual interaction type in their case is quorum sensing and not a metabolic interaction, for example. So yeah, you can we can keep brainstorming like at least for an hour on what else like you can go for in in this thing.
I understand. It's also exciting topic for me too. And so if there's no questions, we can end the session.
Yeah, sure. Thank you very much on the community and all the best in the future for you guys. It was nice meeting you today. Have a nice day.
Thank you for being with us today.
Yeah, sure. See you.